The cell is the structural and functional unit of life. It is the smallest entity possessing the machinery to carry out vital physiological functions autonomously. To maintain internal stability, this microscopic unit must execute a universal set of functions. It must extract nutrients, produce its own energy, eliminate metabolic waste, and divide to ensure continuity. These localized cellular requirements dictate the organization of all multicellular life. Growth, metabolism, and reproduction scale directly upward from this cellular machinery. To create a regulated, stable internal space, the cell requires a hydrophobic boundary to separate the intracellular contents from the extracellular environment. Looking at this cross-section of a lipid bilayer, we see the self-assembly of amphipathic phospholipids. Amphipathic molecules possess both water-attracting and water-repelling properties. Driven by these properties, the hydrophilic heads face outward to interact with the aqueous environment, while the hydrophobic tails point inward, forming a dense central core. This hydrophobic core acts as a highly selective filter. Because the center is made of lipids, it allows fat-soluble molecules like oxygen, carbon dioxide, and alcohol to dissolve into the core and freely diffuse across the boundary. Conversely, the core strictly blocks the passive entry of water-soluble substances. Since they cannot cross the lipid barrier alone, the cell must deploy specialized transport mechanisms to bring them inside. This boundary relies on a precise composition to function. It is built from 40% structural lipids, 55% functional proteins, and 5% carbohydrates. The proteins handle the complex labor. Integral proteins span the entire width of the membrane, providing direct channels for transport and acting as receptors for incoming ligands. Peripheral proteins bind loosely to the surface, performing localized enzymatic activity and structural support. The outer surface features the glycocalyx. This is a carbohydrate layer attached to proteins and lipids that allow cells to recognize signaling molecules and adhere to neighboring cells, an absolute requirement for tissue formation. The structure operates under the fluid mosaic model. The barrier is not a rigid wall. It is a dynamic, fluid matrix where the lipid base allows the embedded proteins to move laterally across the surface, adapting to functional demands. The cell membrane operates as a highly regulated, mathematically precise gateway. It controls exactly what enters and exits, actively dictating the chemical potential of the entire cell. Inside the membrane sits the cytoplasm. Composed of 80% water, this jelly-like material provides the mandatory aqueous medium required to support and facilitate all intracellular biochemical reactions. Suspended within this medium is the cell's largest organelle, the nucleus. It serves as the master control center, housing the primary genetic repository that dictates all cellular activity. This cutaway view of the nucleus reveals the nuclear envelope. It features a strict double-layered membrane required to tightly control the exchange of macromolecules between the internal genetic material and the surrounding cytoplasm. Inside, DNA is stored as chromatin. This material carries the exact genetic code required to regulate protein synthesis and control cellular replication. At the center lies the nucleolus. This dense structure synthesizes ribosomal RNA, initiating the assembly of the very machines that will read the cell's genetic instructions. By utilizing a double-layered membrane, the nucleus successfully isolates and protects the cell's master hereditary blueprint from the highly reactive metabolic chaos occurring in the cytoplasm. Synthesized ribosomal RNA exits the nucleus and enters the cytoplasm to form fully functioning ribosomes. The primary physiological function of these ribosomes is translation. They read messenger RNA instructions and synthesize functional polypeptide chains. Polypeptide chains are linear sequences of amino acids that eventually fold into working proteins. This intracellular flow map traces the path of these molecules through the endoplasmic reticulum, or ER. The ER is a contiguous network of membranes connecting the nucleus to the broader cellular environment. The rough ER is defined by the ribosomes attached to its surface. As these ribosomes synthesize polypeptide chains, the rough ER folds them into functional secretory proteins. Adjacent to this is the smooth ER. It lacks ribosomes entirely. Instead, it is dedicated to metabolic roles, specifically the synthesis of lipids and the detoxification of cellular byproducts. Once synthesized, these proteins are transferred sequentially from the ER to the Golgi apparatus. The Golgi apparatus acts as a central sorting facility. It performs precise structural biochemical modifications, labels the final products, and packages them into specialized vesicles for transport. These packaged vesicles face two final outcomes. They are either targeted for intracellular delivery to maintain other organelles, or they are slated for exocytosis. Exocytosis is the process where a vesicle fuses with the cell membrane to release its contents outward into the body. This physical continuity is absolute. The sequential pipeline connecting the nucleus, the ER, and the Golgi must remain unbroken to execute macroscopic physiological outcomes, like the timed secretion of hormones into the bloodstream. This cross-section of a mitochondrion highlights the sole site of oxidative phosphorylation in the cell. Here, the organelle continually converts raw nutrients into ATP. This chemical energy is the strict fuel source required to power all active cellular processes, including tissue growth and structural repair. Mitochondria possess a unique autonomy. They contain their own independent mitochondrial DNA, allowing them to replicate on their own to meet increasing metabolic demands. They also control cellular lifespans. By releasing the protein cytochrome C, mitochondria trigger apoptosis. Apoptosis is a highly regulated mechanism of programmed cell death, used to safely dismantle damaged cells. Managing the debris of this cellular activity requires lysosomes. These specialized compartments carry a dense payload of hydrolytic enzymes. Hydrolytic enzymes are destructive proteins that use water to sever and break down complex chemical bonds. Lysosomes execute two distinct functions. Through heterophagy, they digest and destroy external invaders like bacteria. Through autophagy, they systematically degrade and recycle the cell's own worn-out organelles. Toxins dictate another specific compartment, peroxisomes. These vesicles isolate oxidative enzymes required for the neutralization of cellular poisons and the execution of beta oxidation. Beta oxidation is a metabolic process that dismantles fatty acid molecules to extract additional energy. These highly volatile, degradative, and oxidative reactions cannot occur freely in the cytoplasm. They must be strictly sequestered within these distinct vesicles to prevent total cellular self-destruction. As we sweep through this fully assembled cell, we see the cytoskeleton. This is the final architectural necessity. Without it, organelles would float randomly in the cytosol. The cytoskeleton is constructed from structural proteins, specifically actin and microtubules, which lock together to maintain rigid cellular morphology and physical stability. Beyond static support, this matrix generates intracellular motility. The protein filaments act as a three-dimensional transit network, providing the exact physical tracks required for the vesicular transport we tracked earlier. This completes the eukaryotic model. Eukaryotic cells are defined by possessing a distinct membrane-bound nucleus. The lipid membrane regulates the environment. The nucleus directs all synthesis. The endomembrane system routes macromolecules, and mitochondria power the entire grid. All macroscopic human physiology, every heartbeat, every breath, and every thought, relies on this structure. It is the direct mathematical result of microscopic compartmentalization.